Shankar, who was studied in India and traveled many countries and done the postdoctoral fellow from the different universities, and he is to guide our students about the opportunities for the pharma students in the world. We have selected him as the resource person because he is one such a sincere and dedicated way. Because of his hard work, he could reach this level. Presently, he is working as a scientist in the Sidra Medicine, Doha, and at present he is staying in the Mexico. His time is early morning, 3 o'clock. So almost in the midnight, he is waiting for us. Only thing, the student-teacher relationship, what we have. About 20 years back, I taught him B Pharmacy, pharmaceutical organic chemistry. But still he remembers and he would wish to do something for the student and his students. Again, we feel thankful for him for sparing his time in this midnight. And with these few words, I request the organizers to say a few introduction about him and start the program.
Thank you, sir. Now we introduce our scientific session for research person. Dr. Selvasankar Murugesan is a research scientist at Sidra Medicine, National Institute of Pharmaceutical Education and Research, Doha, Qatar. And sir has done a postdoctoral fellow at Sinivasta, doctorate in science, and sir did his bachelor's in pharmacy at National Institute of Pharmaceutical Education and Research. And sir has licenses and certification in data visualization for data analysis and analytics. And sir has in skills of DNA sequencing and molecular biology. So with this few introduction, I request sir to hand over the session.
Okay. Yeah. Can I start, sir?
Yes, sir. Yes, sir. You can start.
Yeah. First of all, I want to thank Dr. Alagasamy for this opportunity to share my experience. Let me share the screen. I hope can everyone see the screen? Right. Okay, yeah. So as Dr. Arasami mentioned, today I'm going to give just kind of whatever the experience I have gone through in our pharma career. So what are the research opportunities what we can get from the countries I have been through? This is what in today's talk I'm going to share. So in this, before that, I want to talk something about overall in pharmacy research. Mainly, our role is to work on any kind of new medically active ingredients, which is called as drug, and to improve their formulation by changing their formulation or the increasing the efficacy, and also based on the suitable body site to improve the drug delivery. This is what mainly focusing on, and there are different types of research in this, particularly basic research, applied research, and clinical trials and translational research. If we come through in the last few decades, whatever the main important breakthroughs that is happened mainly for the medicinal field, that is through pharmaceutical research only. There are some few examples in the current, even the last during the COVID, the vaccines, and many other novel medicines, which is in the recent time we are trying to identify through the artificial intelligence, and with that we can find the novel molecules and their role, how that can be a new novel drug molecules. So in the basic research, mainly we focus on what is our fundamental mechanical question, and what what are the steps we need to answer that question. In the applied research, how we can improve the whatever the medically active ingredient we found, how we can improve the what kind of formulations can improve the efficacy of the drug. In the clinical trials, mainly we focus on the safety for the patients and how much it can be effective when it is applicable to the any kind of treatment for any diseases. In the translational research, where it's forming a bridge between all these three clinical trial, applied research, and basic research. So how the from the lab discovery to the market to come through this. So translational research can focus on that. So in this pharma research, what are the careers? If you focus on the academic research position, where you can go for the mainly like professors and other things, in the teachers, where you can go for lecturers, and particularly in kind of research, where you can mentor students and focus on their career growth by teaching them the research activities, giving them the project goals to attain, to meet up to the task, and teach them the regular whatever the basic research activities for their project need. In government sectors, for example, for the drug formulation and for the pharmacopoeia things, you can go for the drug evaluation purpose, and the public sector government organizations. And in case of pharmacovigilance, you can go for the drug regulatory affairs and the other things. Yeah, I saw the program. I think in the previous talks or in the upcoming talks, you will see more brief about this. In this talk today, we will be focusing what are the currently available research opportunities once you complete your B Pharm, and this is what I'm going to share. When you focus in pharma industry, geographically mainly, these are the United States and some European countries, particularly Germany, Switzerland, and Belgium. They are one of the huge countries where they invested a lot to improve the innovative and novel drugs and to improve the drug development. These are the countries which huge manpower and the facilities to bring the novel drugs to the market. And in the last few decades, you see our Asian countries and Africa, they are one of the huge market for the drug development and the clinical trials. In the recent times, there are several countries, they are investing a lot to focus more towards the novel drugs. So that's why in pharma industries, it is always, there is a huge amount of growth. And at the same time, you will face many kind of challenges. For example, if you come into the research activities, one of the main hurdle is to get the funding. So in the past few decades, as you know, because of the economic recessions, there are a lot of limitations. And also in the regulatory hurdles, like many documentations, ethical approval, these are the other things which can slow your process. So all these, basing all these hurdles, you need to give your work on time. That is, the time limit is another hurdle which normally the researchers they face. So directly I will come through since I have done my PhD and everything. So I can share some of my experience in Mexico. What kind of research opportunities, mainly focusing on the pharmaceutical sciences. So in that, I can say one of the big funding agency in Mexico is CONACYT. This is similar to what we have in India, like Science and Department of Science and Technology, or CSIR. So they have the funding agency not only for the local students and also for the international students. So you can come through like a collaborative from India and Mexico, or directly you can come to Mexico as a PhD or for your masters. So mainly their aim is to focus the now innovative research, focusing on the novel pharmaceutical drugs. So for pharmaceutical research-based institutes, there are these 4 institutes, which is most well-renowned in all the Latin American countries and also in the North America. So if you see Sinvesta, UNAM, and Institute of Polytechnic National, which is one of the, in English you can say Polytechnic National Institute, and Mexican Institute of Social Security. These all four institutes, they mainly work on the pharmaceutical research, particularly Sinvesta, they mainly focus on through pharmaceutical chemistry and biotechnology, they focus on the drug discovery, and also through pharmacology department, they will focus on the pharmacological effect and the other kind of experiments. In UNAM, mainly they focus on the lab, similar to Sinvesta, on the drug research. If you focus on the Polytechnic National Institutes, they mainly focus on the policy making and also the regulatory affairs, including the pharmacovigilance and pharmacy practicing. And similarly, IMS also doing the same activity. In these four institutes, you can easily, after your bachelor's, if you want to continue for the master's, with a good GPA or CGPA, where if your scale is 10, then for the 10, if you have out of 8, then you are eligible for the scholarship based on the institutes. Sometimes you need to go for the some coursework. In case of PhD, you can directly go for the you will directly get the scholarship upon clearing a TOEFL, or in some institutes just with your master's thesis, you are just with a small presentation you can directly apply for the PhD program. So if you see in Mexico, there are many pharmaceutical industries where they focus on the novel drugs, and also they work on the multiple clinical trials. If you see in the last decade how the huge amount of investment is growing, which indicates there are always in the need of pharmaceutical research oriented peoples are in need in Mexico. And also in the upcoming slides, I'm going to share some of the research opportunities which you can find in Qatar, where I'm currently working now. So in Qatar, so in the current Emir's goal, by 2030, they want to focus all the areas, particularly in the pharmaceutical research, by focusing in the not only pharmaceutical research and also in the healthcare system, in all the hospitals. They want to develop all the Qatari citizens, and they want to be in a fulfilled way where they can make their own research without importing any for medicaments from the other countries. So particularly they focus on the drug discovery and also the personalized medicine through the recently launched Qatar Genome Program. And also in the same Qatar Genome Program, they are also working on the pharmacogenomics, based on how the genetically variants, how they differ to the different pharmaceutical drugs. And they also currently they are going to focus on the clinical trials. So by in the Qatar's main aim is to, instead of importing the pharmaceuticals, they want to focus on the local drug development. So the main key institutes in Qatar focusing on the pharmaceutical research is through Qatar Foundation, where they have the different universities. Through that universities, they have covered the healthcare and the biotechnology and the pharmacy. Through Qatar University, they have a specific department for pharmacy, where they focus on the pharmaceutical based research. In addition to that, Hamad Medical Corporation, where they do all this clinical trial, and by collaborating with many US and European institutes, and they are working on many multiple drug trials. In Sidra Medicine, the institute where I am working now, they mainly focus on the Qatar Genome Project based. So by characterizing all the genetic information, they want to focus the specific precision medicine. And also the institute is focusing on the mainly on women and the newborn children health. So they want to avoid any kind of pregnancy associated complications. So Sidra is mainly working on that. In Whale Cornell Medicine, they mainly covers all the biomedical sciences and also pharmaceutical research. They mainly working on the university based program, And part of it, they have their research program, which is covering pharmaceutical research as well. Apart from that, the recently, like 5 years back, they launched Qatar Biomedical Research Institute, where they cover cancer research and also the diabetes. In addition to the pharmaceutical sciences, they also focus on how the microbiome in how it's working with pharmaceutical drugs, how it is interfering with the drug metabolism, and so in some cases it can enhance the pharmacological effect. In some cases, it will block the pharmacological effect. So they are focusing on the mainly these studies. So for the fundings, so similar to in India, how I mentioned the CSIR and DST, in Qatar, the main funding organization is Qatar National Research Funding Agency. They have the grants for the investigators, for the masters and the PhD program. They have called program called Europe and JESRA. And for the scientists, there are multiple programs, and one of the important program called National Priority Research Program, where they cover one of the commonly in the last few decades autism and some other genetic complications. In currently, they are focusing on that kind of research. They want to find what are the possible factors that are lead to cause these kind of genetically um, uh, mentally disorders. And these scholarships are not only for Qatari students, and also they welcome international students to pursue their masters and the doctorate in pharmacy and pharmacology. And in many Qatar universities, the Qatar institutes, they have the tie-up with Harvard and Stanford universities in US and also in UK. So, as I mentioned, Qatar and Mexico, they are mainly focusing. There are many opportunities in private, government, and the academic institutes. So there is a rich demand in clinical researchers and pharmaceutical scientists who are interested on precision medicine and pharmacogenomics to improve your career. So, as I mentioned, the current problem there are in every field. One of the main important hurdle is whenever there is always there will be people will be moving whenever they find the better opportunity, and there are limited sources where it restricts some budget restraint. And in Qatar, their main aim for the in the upcoming years is to make all the Qatarization where they want to develop their own technology and also patent their newly identified molecules. So with this, I want to conclude. So in not only in US and other main European countries, in the recent times, Asia, Africa, and Middle East are also one of the growing hub for the innovative research in the pharmaceutical field. So in Mexico, CONACYT, and in Qatar, QNRF is one of the funding bodies where they provide scholarships for all the international students as well. So with this, there are multiple opportunities you can find in both academic and the pharmaceutical companies in the private sectors. And with all the funding restraint, you can improve your studies. And also by identifying the new novel innovative pharmaceutical drugs, you can improve more pharmaceutical investment and find more opportunities. So these are the the mainly I wanted to share my experience in Qatar and Mexico. There are many opportunities in US and Europe, but in compared to there are many people they used to go, but in Mexico you have a lot of opportunities with a good amount of fund scholarship also. So with this, I want to conclude my talk, and I thanks again Dr. Alagasamy for the opportunity.
And briefly elaborate. Majorly your experiences and ongoing hurdles in state. Thank you so much, sir. And now the session is for interactive session for the students. Kindly, if any interaction you want, please come forward.
Good afternoon, sir. Good, good afternoon, sir. Sir, can you elaborate a little bit on the clinical pharmacy, how to get jobs in Mexico, other than research, if you have in clinical area?
So, yeah, honestly speaking, I don't have much experience on the clinical pharmacy since my after my master's completely my main research is focusing on the human microbiome. So in that way, I'm more into the molecular biology. But in the Mexico, there are a lot of multinational company, even Dr. Reddy's, they have their unit in Mexico. So clinical research, I don't have experience on that, but I know people who came mainly for the this formulative. I mean, pharmaceutics persons. There are many opportunities for the drug formulation and R&D. That kind of work. There are a lot of opportunities in Mexico, but I don't have experience on this clinical pharmacy.
Thank you, sir. Sir, regarding Doha, like after BPharm, many of the students, they would like to work abroad as pharmacist. Some of them are not doing their PG also. They may work, they wish to go abroad in that way. In the Gulf countries, they have some test for that purpose, Ministry of Health.
Yeah, exactly.
So in
So
Doha also we have similar.
yeah, almost all the Middle East countries, when you are in the particularly in the healthcare system, for the pharmacists, they need to clear the Ministry of Health exam. So they need to pass once. After that, they need to attend regularly, like many workshops or to maintain their license. They regularly meet these requirements. For that, after clearing the exams only, they are eligible to work as a pharmacist in Middle East countries, particularly.
Thank you, sir.
Good afternoon, sir. Sir, after family, what are the opportunities for clinical trials in Qatar?
So for clinical trials in Sidra, we have for our own institute itself, we have only a specific department focusing on the clinical trial. So I guess for the PharmD also, if you want to practice, then you need to clear the exam as well. Then you can be as a researcher in our institute as well. I mean, you need to maintain your license, then you are eligible to participate on the clinical research program.
Okay, sir. Thank you.
Good afternoon, sir. Is that a clinical oriented opportunity after PharmD?
I have a colleague. She did her Form D, but initially she find many difficulty into get into the clinical trial. But somewhat in the last year, she was able to clear her exam. And in Qatar, in the last few decades only, they started to work on this clinical research. So in Qatar, maybe in the next decade, definitely there will be huge amount of opportunities. Currently, they are mainly focusing on the Qatar Genome Program. Once they characterize the Qatar Genome Program, maybe they will start working hugely on the clinical trial. Maybe it will open a lot of opportunities.
Thank you, sir.
Yeah.
Good afternoon, sir. Are there any advanced programs after PharmD?
PharmD, it's almost equivalent to doctorate only. So after that, depends on the institutes. Whether you can go for the research oriented means you can directly apply for your postdoctoral fellowship, and from that you can focus either you want to be an investigator or you want to work on the pharmaceutical companies.
Okay, sir. Thank you, sir.
Yeah.
Thank you, Dr. Selvasankar. Is it visible?
Yes, sir. Yes, I can see that.
Okay. So this is wholehearted thankfulness that we are extending from MNR College of Pharmacy for your valuable time that we are extended to our students. So really it was a good lecture, and many students could understand at least some of the new ideas or the new opportunities that they could understand. So thanks a lot for joining and spending your valuable time, Selvasankar.
Thank you so much, sir, for the opportunity.
Okay.
Thank you.
Thank you. Kindly accept this as a token of appreciation from the MNR College of Pharmacy.
Thank you, sir. Thank you.
Okay, ma'am. Thank you, one and all.
Professor, Geetam School of Pharmacy, Hyderabad, for oral presentation.
Good afternoon, everyone. This is Dr. Priya Singh from Geetam School of Pharmacy, Hyderabad campus. Today, I'm going to present my research work that is basically based on the synergistic strike on breast cancer cells. Basically, I'm dealing with the two dual drug-loaded cubosome preparations and their evaluations in the breast cancer management. Turn the next slide, please. Basically, we will start with the breast cancer. What is breast cancer? We already know. And this is the second most leading cancer which is growing in the world right now. And major challenges is they're developing the resistance. Whatever monotherapy we are developing, whatever the treatment we are having right now in the market, they are developing the resistance. The cells are developing resistance in it. So there is a one solution can be over the monotherapy can be the dual drug treatment. Means we are giving the combination therapy to the breast cancer cells to treat that and do not develop the resistance, and they can cure better. For that, we have chosen two different drugs, and we are preparing a drug delivery system, basically the nanotechnology we are talking about here. The cubosomes, the nanocubosomes that we already know, nano means the 1 to 100 nanometer size range or a 500 nanometer size range is considered as a nano formulation here. So basically, we are using this drug delivery system to manage the breast cancer treatment here. Can you go to the next slide, please? Okay, so in my study, the objective of this particular research work is, in the first, we need to develop the nanocubosomes which loaded with two different drugs. And these two different drugs are working on a different receptors. First one is a bosutinib, second one is a exemestane. They are working in a different receptors. So we can treat easily and they cannot develop the resistance easily to the breast cancer cells. Second, after preparing the nanocubosomes, we need to optimize the formulations by the QBD design, quality by design. Next, to evaluate their parameters. We have prepared the nano formulation, we have optimized, but what next? There should be evaluation parameters. Their particle size, drug loading efficacy. How will you evaluate that your drug delivery system or their nano formulation is working properly, or they have a better as compared to the conventional therapy? So we have to do the evaluation. After that, we have done the in vitro, now come to the cell line studies. That is also in vitro studies, but we are targeting the cells, cancer cells. Because we have to see the treatment, the effect of these particular formulations on the cancer cells. So we have chosen two different cancer cells, that is MCF7 and MDMB231. That is our breast cancer cells. And we have checked the cytotoxicity efficacy on these particular cells by preparing these nanocubosomes here. Can you go for the next slide, please? Okay. The first question will come in mind, why nanocubosomes? Why cubosomes I have chosen, right? I'm talking about combination therapy. We are loading the two different drugs, right? So we need more capacity to load. We can do it with the liposomes also, nanoparticles also, but why we have chosen the cubosomes? Because they have more drug loading capacity in it. Why? Because they have a cubical structure, the cubes. So the drug can be entrapped or encapsulate in the corners as well. So that's why we have chosen this particular drug delivery system to load these drugs and to see the effect on the breast cancer cells properly. Then there is one more thing, their cellular uptake, means there is a lipidic nano formulation, so their uptake inside the cancer cells will be easy and their internalization will be good. That's why we have chosen this particular drug delivery system to treat the breast cancer cells. Now come to the formulation development. We already know there are so many formulation we have to prepare to get a one optimized formulation here. So by using the one software, the design expert, we can do it easily, right? So that's why we have using this design expert software to optimize our formulation. And we are getting that by the software, the predicted value of the GMO will be, for your information, GMO will be your lipid. And why we are using GMO? Because they have a property that when it comes into contact with the water phase or aqueous phase, they will create a cubes-like structure. That's why specifically we are using GMO here. Poloxamer, 80 mg, sonication time will be 15 minutes. So the software is predicted your particle size will be 336.41 nanometer. The software will telling us. Entrapment efficiency will be 88%. But when I am formulating this particular formulation in the lab, and after that I'm getting the value of particle size, that is 297.4 nanometer, and the encapsulation efficiency will be 92.5. So predicted and whatever we have observed after preparing that particular ratio. So that is your formulation development and optimization part. By using this software, we have optimized, we have seen the different ratio of how it is affecting the particle size as well as the encapsulation efficiency. Next slide, please. After the optimization, we got one optimized formulation. After that, we have done the multiple characterization. First one, particle size, PDI, zeta, loading, entrapment efficiency. Also, we have done the morphological studies like SEM, TEM evaluations. Percent drug release, how much drug they have released in 24 hours. Antioxidant studies, stability studies, and cell viability studies on the MCF7 and MDMB 231. And in the last, we have done the cellular uptake studies as well. So now I'm coming to the first evaluation, that is particle size. Particle size, we can see that after optimizing that particular formulation, we are getting 297.4 nanometer size range. And also we are getting the zeta minus 29.13. And also we can see the last two C and D figures basically represented our SEM and TEM evaluations. The first one, the C, it is our SEM image that clearly shown that cubic structure of the cubosomes. By seeing, we can see that this is your cubosome because the cubes are clearly visible here. Also, we can see in the figure D, that is our TEM evaluation. In that, we can see the internal structure also. That is the cubic particles we can see, but also we can see the internal structure of the particles here. So, by doing this SEM and TEM evaluations, we can say that our nanocubosomes has been formed here. Next slide, please. Okay, now come to the powder XRD analysis. As we already has discussed in one of our scientific session that crystalline has a more stable in nature. So to check that particular nature of our cubosomes, their APIs and excipients, we have done it the powder XRD analysis. In that we can see that first and second one will be our API only. There is no any formulation, no excipients is there. But in case of third and fourth, the red one will represent your physical mixture. Physical mixture, we are taking all the excipients and just mix it together. That is your physical mixture. And in the last, we can say that there is a formulation, the nanocubosomes that we are talking about here. So it is comparatively that physical mixture, it is showing more crystallinity. Means there are more stable in nature. So that crystalline structure, cubic structure we are talking about. So we can say that they are having a more stability as compared to the free drugs or a API. Next, please. Okay, now move on to the next, that is your in vitro drug release study. Any formulation, when we are doing tablet, capsule, anything that we have to do the drug release. Ki how much drug is releases from the formulations here. So firstly, we have done it for the 24 hours in PS 6.6. So we can see that there should not be a black background, but it's okay. In case of the green, we can see that that is our formulation, nanocubosomes, and blue one represents the free drug, that API only. We have put it the API directly and see the drug release. But in case of cubosomes, you can see that the control and sustained release pattern of the release here. So that nanocubosomes basically representing our control and sustained release manner as well. That is very useful for the cancer therapy or to not developing the resistance and to showing the control and sustained release profile for the drug as well. Next one is your antioxidant activity. In that we can see that there is two things we have compared. The first one is a API, combination of API and nanocubosomes that is loaded with the two API that we have talked about. So in case of the API, it is showing the lesser result as compared to the cubosomes. Because cubosomes have some cubes, they have particular release. That's why they have a nano size range. So they are showing the better antioxidant activity as well. They have a better scavenging activity. So that's why we can say that our formulation that we have developed that is better than the free drug or a API or a single drug loaded formulations. Next, please. Okay, in the next slide, we had just talked about the in vitro cell line studies here. In case of, in the first graph, you can see that percent cell viability studies. In that, we have seen the different concentrations we have taken of the our drug and formulation and API, and we have tested on this particular cells. And we have seen that if we are increasing the concentration of the nanocubosomes, it shows the lesser percent viability here. Viability means the lesser number of viable cells are present in that. Means our formulation is killing the cancers more as compared to the free API. What can be the reason behind it? That can be because of the proper internalization of that particular cubosomes inside the cells, and also they have a better release in that. That's why. And the second one is your IC50 values of the treatment. In that we have compared with the second last one is your nanocubosomes formulation that is shown the minimum IC50. Means we have to give this much of concentration to kill the 50% of cells from that particular sample. So that result shown by the percent viability assay, that is MTT assay as well. Okay, the next slide, move on to the next, that is cellular uptake. Means how effectively cell is taken up that particular formulation, that can be seen in this particular figures. That is a confocal microscopy of the formulation of the MCF7 I have mentioned here as well. In case of, we have loaded our formulation with the fluorescent dye, that is rhodamine and FITC. In case of that formulation, you can see that the red one, that is your rhodamine, and green one in the middle, that is your FITC. So we can clearly see that in that cell, there is a better uptake of the nanoparticles inside that. We can say that our formulation is easily going to be uptake by the cancerous cells and causes apoptosis in that. So that is the cellular uptake in case of MCF7 we have talked about. Next, please. In the second one, that is another cell lines, that is also a breast cancer cell lines, that is MDMB231. We have done it only with the FITC, but when we are comparing with the control API, free drug, and also the nanocubosomes, so we can clearly see that this particular free drug has a lesser uptake as compared to the nanocubosomes. This is also a confocal microscopy image that we have seen that uptake of free drug is lesser as compared to the nanocubosomes. So because of their cuboidal stability, nano size range, they have better uptake in the cancerous cells as well. Next, please. These are the stability studies. We have kept it in a different conditions, that formulation, and we have evaluated again for their particle size and encapsulation efficiency. That is if there is a major variation in the result or not. So in that case, we have kept it for 0, 30, 60 and 90 days, and we have kept it at two different temperature, that is 25 room temperature and 4 degree in freezer we have kept it. So, and we are comparing the percent evaluation, encapsulation efficiency and also the particle size. So we can see that there is not major variation comes in after 90 days, we can see that. So we can prove that that is also a stable formulation as compared to the free drug as well. Next slide, please. So basically in this research work that whatever we have till done, that is basically successfully we have formulated this formulation and achieved that whatever particle size we need and encapsulation drug loading. And also efficiently we demonstrated what is cellular uptake should be. And also we have see that the nanocubosomes have a superior cytotoxicity activity as compared to the single loaded drugs, and also the APIs. Exhibit sustained release we have seen into the graph, and also they can potential for overcoming the drug resistance, because we are mentioning the two different drugs here, and they are both are the targeting to the different receptors here. Next, please. In conclusion, our study has demonstrated that this is a effective therapy can be in case of the breast cancer treatment, and it is effective moreover the monotherapy because of this is overcoming the resistance problem in the cells. So basically, for the further evaluation, we need to do the in vivo to validate the data that it's exactly or literally it is working or not. So that is the project work that we have done. Next, please. I would like to acknowledge the Geetham Visakhapatnam campus for Murthy SAF facility that we are given with the SEM and nanoparticle size analyzer, and AIMS for the TEM analysis, and NCCS Pune for the supplying the cell lines, that is MCF7 and MDMB. And this work has been funded by the Geetham Research Seed Grant. So thank you so much everyone. And thank you MNR College of Pharmacy for giving me this opportunity as well. Thank you so much for that.
Ma'am,
Yes,
I have one question.
Yes, sir.
Why you have chosen that two cell lines in your study?
Sir, we just want to check that because we have using two drugs, sir, bosutinib and exemestane. They both are working into the different receptors here. So basically, MCF7 that we are saying, that is a estrogen positive receptor cells, we can say that. So in that case, we are just using both the cell lines to see that both the in both the cell lines is our formulation is working, or only one drug is working on that particular cell line. So that's why we have chosen two different cell lines to prove that this is literally working if you are loading two combination drugs here.
Okay, ma'am.
Madam, can you elaborate on the preparation method once again?
Sorry, ma'am.
Method of preparation once, can you elaborate?
Okay, for nanocubosomes.
Yes, ma'am.
Actually, we have to take the lipid, that is GMO we have taken, and 120 mg, and we have dissolved into the acetone or ethanol, whatever organic solvent we can take, and the drug has to be dissolved in that only. And in the water, we have to drop wise, we have to do it. That is your hydrotope method of the nanocubosomes, and then we have to go for the sonication. And after that, whatever the particle dispersion we are getting, that is your nanocubosomes, we can say. That is hydrotope method we have used to prepare this nanocubosome.
Immediately after the preparation, what is the immediate test which you will be performing so that we can confirm that the product is formed?
Basically, immediately you need to sonicate. Means if you thought ki this is the overnight procedure, we have to kept it for stirring for 24 hours. If you are not doing the sonication after 24 hours, it will make a cluster. Won't form the cubosomes. So that is the major important part that you have to do, that is do the sonication after preparing that. After completing the 24 hours on the stirring, you need to do the sonication first. Otherwise, you will get bigger particle as well.
One more question, ma'am.
Yes.
You said it is an effective.
Yeah.
So is there any confirmatory test?
Sorry.
Confirmatory test.
Confirmative.
So how you will say it is showing an efficacy, good efficacy?
So we have done the MTT also, cytotoxicity for that, cellular uptake. But that further evaluation we can do for we have to go for the in vivo. That is our future research project, that we'll do the in vivo for that particular formulation as well. We have done it till only the in vitro cell line studies. And by seeing that cell viability studies and cellular uptake, we can say that it is effective in case of cancer cells. So that's why we can say.
So can we do any biopsy testing in vivo?
So definitely we can do, sir, but that is in a future perspective, that we can future evaluations we can do for the preclinical stage all.
Okay, thank you.
Thank you. Done?
So next scientific session will be there. So e-posters will be held at seminar hall, third floor, and followed by oral presentations. According to the department, it was scheduled, and first is Department of Chemistry and Intellectual Property Rights and Pharmaceutical Analysis and Pharmacognosy. These 4 departments will be at first floor, first PharmD classroom. And then followed by a pharmacology and pharmacy practice department will be at a second floor, third PharmD classroom. Then followed by a pharmaceutics department, third floor, fourth PharmD classroom. And scientific presentations includes a pharmaceutical chemistry, drug design and discovery, pharmaceutics and drug delivery, pharmacology and biotechnology, pharmacy practice and clinical pharmacy, pharmacognosy and phytochemistry, pharmaceutical analysis and regulatory affairs, intellectual property rights. And now next oral presentation is Kirtana Shri from Pratishta Institute of Pharmaceutical Sciences from Suryapet.
Good afternoon, everyone. This is Kirtana Shri from Family 4th year from Pratishtha Institute of Pharmaceutical Sciences, Suryapet. Today, I want to explain about cryptic pregnancy. Title is Cryptic Pregnancy in an Evolutionary and Clinical Perspective, and subtitle is Understanding Denial of Pregnancy Through Biological and Ethical Lenses. Introduction. Pregnancy is usually involves in physical, emotional, psychological adaptations. However, in this cryptic pregnancy, you can observe that subset of women will deny their pregnancy cautiously or subconsciously, and the term of cryptic pregnancies remain undetected. They are pregnant, but they don't know that they are pregnant. It may risks for both fetus and also mother. In cryptic pregnancy, it's a condition where woman is unaware due to lack of symptoms, and here we can observe the incidence and epidemiology is among 475 cases, we can observe one case of cryptic pregnancy. Often it undergoes, it unrecognized due to lack of symptoms like putting up weight or any type of baby bumps. No bump is present in these cases. Baby grow inwards, like not out. And minimal weight gain and fetal movements. There will be no fetal movements, there will be no weight gain. And in this case, periods are regular and irregular. Types of denial. Psychotic denial associate with mental illness and schizophrenia. They may, non-psychotic denial, pervasive, total, unaware. In this non-psychotic, in psychotic, they may face schizophrenia in this condition, and non-psychotic, total, unaware. They don't know whether the fetus is present in them or not, and affective acknowledgement without any emotional connection. They'll be not emotionally connected to the fetus growth, and they'll not take, there'll be no parental care, they'll not take care of that fetus growth. And persistence, realization in third trimester. They may know about their pregnancy slowly after 8th month or 6th month of time period. And in evolutionary perspective, cryptic pregnancy refers the condition biologically. Here, this may cause due to the genomic imprinting disruptions and spontaneous when the mother have frequent abortions. This cryptic pregnancy may occur. Due to low quality of fetus formation and forced cooperation, which is stressfully from family pressures or partner pressures. Psychologically, they'll be very depressed, and in these cases also, they may cause cryptic pregnancy. Here we mentioned 3 cases. We filtered out and mentioned 3 cases here. One is like surprise delivery in an emergency department. Already the patient was recently delivered a baby, and this was her second pregnancy, which was unknown, and already her vagina was little bit stretched, so she delivered a baby in a bathroom. And case 2, emergency delivery in California emergency department. Due to frequent fetus loss, due to frequent abortions, suddenly she delivered a baby unknowingly. Case 3, suspected preeclampsia with unknown pregnancy. In this case, the fetus was observed in 6th month of her trimester, and in this 6th month, the baby was truly unhealthy, so they have to remove the fetus. But the parent is willing to keep, but the fetus was too weak, so they have to remove. Here, biological mechanism, we can't detect this cryptic pregnancy by common pregnancy tests due to low level of HCGs and hormonal imbalances, etc. And clinical challenges, there will be no parental care, as I already mentioned, and surprise deliveries, which are unsafe settings, which we observed in case 1. They'll have severe stomach pain. They'll go to emergency department due to stomach pain, but they deliver a baby. It was like a surprise party at 12 o'clock. And emotional trauma, increased neonatal risk. And in these cases, the babies may born with meconium-stained babies. Meconium-stained baby is nothing but the amniotic fluid in the belly will be mixed with the poop of the fetus. And also distress. Here the ethical and legal dilemmas are anatomy, and why I mention here ethical is the mother is unaware that she is pregnant. So here they inform the consent. They have to take the consent of the patient whether she is ready to deliver physically and emotionally, and it can't be detained under according to Mental Health Act. The obstetrician and a legal department member and a psychiatric team have to sit with the patient, and which is like mother, and they have to discuss about her pregnancy, sudden delivery, and they have to stable that mother psychologically, and need to multidisciplinary ethical consultations. They have to consult her frequently. They have to check over her physical health and psychological health also. Here diagnosis and management, we can't give any type of management to her, so because she delivered a baby already, so we can't do anything. We have to take care after delivery. We have to take care of baby. Here high suspension, high risk women, and only ultrasonography and frequent HCG testing will come to know about this cryptic pregnancy. Psychiatric evaluation for denial behaviors. They'll have at 6th or 8th time of period, it will be a very surprise to mother to deliver a baby, or to know that she had a fetus in her, and so the psychiatric sessions should be take frequently to the mother, and they have to take the permission of both the parents that they are okay or not. According to Mental Health Act, they have 100% legal chances to adopt, to give the adoption. If they are not willing to keep the baby, they can give for adoption legally, and supportive and respectful counselings. Here proposed classification is negated pregnancy. They are willing and the baby is also willing. Both are in a healthy conditions. And denied, complete lack of awareness. They are completely lack of awareness until delivery. In negated pregnancy, they'll be aware at 6th or 8th month of period, and consoled, aware but hides. This may be in the having baby illegally. They'll know that they have a baby, but they cover up all that. Further subclassifications are according to gestational stages, or whether it's 6th or 8th or 9th. Here the conclusion is we have to know that cryptic pregnancy is also a type of pregnancy which we'll see in most of the cases. Among 475 cases, we'll see one. So it's not a rare as we assumed. And it blends biologically. It also blends like common pregnancy biologically, ethically, psychologically. Awareness and sensitive handling is crucial. We have to be very aware of that, what the condition we are facing and what the condition of the baby inside us. And the maternal and fetal outcomes should be good. This new terminology and this new research and this new study may help the medical professionals nowadays for easy detection, for easy diagnosis, and for easy management. Thank you.
What is the difference between a cryptic pregnancy and an ectopic pregnancy?
Actually, it's a type of cryptic pregnancy. It's a stage of cryptic pregnancy. Cryptic pregnancy is nothing but we are unaware. We will not know that the fetus is present. It may be legally or illegally we can have a baby, but it is unaware. Until we deliver a baby, we can't know that the fetus is present. So commonly we'll have common symptoms in pregnancy like vomitings, putting up some weight, or having a belly. In this cases, no. Even if we go through the pregnancy test, it will get negative due to low HCG levels.
So what might be the reason for low HCG?
Hormonal fluctuations and improper hormone release, and also like genomic imprinting disruptions of maybe the gene of mother or maybe the gene of father is a little bit disrupted in imprinting, and low HCG levels maybe the patient was with PCOD or PCOS issue.
So is there any case you have seen in hospital?
Actually, it's my review paper, sir. I just saw it in an Instagram and I felt to have a review on that, and I researched all the review papers. Up until now, I haven't seen, in my hospital, I haven't seen these cases.
So is there any relation between an amniotic fluid and low birth weight?
Actually, in these cases, the baby will, 95% of babies will be very healthy. But I here mentioned the amniotic fluid is like, in some cases, the baby will give birth with the poop in the amniotic fluid. It will mix in the amniotic fluid and baby will deliver with that. So I mentioned amniotic here.
So is there any complications with meconium?
We have to go through, it's a different topic.
Okay. So next presentation, Vishnu Priya. Vishnu Priya from Arya College of Pharmacy.
Good afternoon, one and all. And I'm Vishnu Priya, B Pharmacy third year from Arya College of Pharmacy. And today I'm here to present you about the post-COVID effects on heart and blood. We are all aware that COVID-19 was a global pandemic in the year 2020. And it's been more than 3 years, so that it has given a lot of disorders in our body that led to cardiovascular diseases more. Some people have gone through many respiratory illness, but it was mild to moderate. But more than that, we are seeing nowadays cardiovascular diseases have increased a lot. So, coming to the heart and blood related issues. When the virus invades our body releases inflammation and cytokine production to overcome the virus. So, the inflammation is useful in small amounts, but due to the over-inflammation, we may observe heart arrhythmia. Due to the inflammation, it may cause arrhythmia, that is losing the electrical signals of the heart. And next, the myocarditis. Myocarditis is inflammation of the heart muscle. And next, the pericarditis. It is the inflammation of the membrane around the heart. And heart failure. Heart failure is due to the fluid buildup in the lungs and body. And next, the thrombosis. Due to the inflammation in the blood vessels, the blood clots are formed, and that may lead to stroke. And next, ischemic heart failure. It is due to the changes in the heart muscles that may weaken the heart and cause the arrhythmia or any stroke. And next, who are at risk? Nowadays, we observe many of the people are at risk, like example, diabetic patients and the people with pre-existing conditions like hypertension and obesity and coronary artery diseases and many other people. And it may also lead risk in the pregnant women. Pregnant women may also have the effect of this heart and blood related issues due to the post-COVID effects. And next, how can we prevent and manage this post-COVID effects? First of all, we have to turn our lifestyle into, we have to modify our lifestyles and changes in the diet. And following the fitness and everything can help in management. And secondly, adhering to the prescribed medications. Before that, we have to undergo regular checkups. These regular checkups helps us to know about the condition of our body, and so that we can understand what is the problem and how can we overcome it through the medication. And finally, I conclude that to build a good future, a resilient future, we have to focus on our health. Health is very important, and in that, heart is the main. So we have to concentrate our health, and we have to manage everything and overcome this cardiovascular diseases. Thank you.
Check. Is there any diagnostic parameters to avoid a post-COVID effect?
Diagnosis is that we have to go for regular checkups, sir, so that they'll suggest the proper medication based on the condition of our bodies.
What about parameters?
Sir?
Diagnosis. Tropi, tropty.
Sorry.
So if we want to reduce on post-COVID effects, is there any diagnostic parameters? Can we find out any blood test or else any angiogram?
Heart checkups and everything, and examination of the blood vessels, so that we can understand any types of diseases.
So what will be the management?
Management, we have to take the prescribed medications, and next changing our lifestyles, and then undergoing regular checkups.
So blood related means how we can change our lifestyle.
Blood related. Taking proper diet, sir, and following some exercises and everything can help to overcome this.
Okay.
Thank you, sir.
Next presentation by Jasmita from Arya College of Pharmacy.
Good afternoon, everyone. I am Jasmita from Arya College of Pharmacy, a third year B.Pharm student. And today I am very excited to tell you all people about the bite therapy. What is the bite therapy? The bite therapy showing T cells where to punch the cellular villains. What is cellular villains? The cellular villains are the cancer cells which are developing in the body. So firstly, introduction to bite therapy. BITE stands for bite-specific T-cell engager therapy. So we all know that chemotherapy is the very traditional and conventional way of immunotherapy. But when it comes to bite therapy is the most advanced and modernized therapy to treat the cancers, and it is the most and the bite therapy got the most promising results. So how bite therapy works? So bite molecules has two arms, where one arm binds to CD3, that is, one arm binds to CD3 and another arm binds to CD19. So one arm binds to T cells and another arm binds to cancer cells. Eventually, in chemotherapy, T cells are subjected to kill the cancer cells, and in chemotherapy as well as cancer cells along with healthier cells which are growing in our body are killed. But when it comes to BiTE therapy, the T cells are subjected only to kill the specific cancer cells. So the BiTE therapy itself, the name the name tells that bispecific. So what is bispecific? T cells are specified only to kill the cancer cells. In BiTE therapy, no healthier cells are killed. In this preview, healthier cells comes under hair follicles, nail follicles, and all other examples. So next come to the history. In 1960, the concept of BiTE is introduced, and after that, slowly the development started, and in 1985, the concept of BiTE therapy developed, and the BiTE molecules also developed, and there is a drug named blimetamomab. So after that, in 2012, Amgen University, according to the article 221, the Amgen University has acquired the Micromet. Micromet is the company which is located in Germany. That is the company which developed the blimetamomab drug, which is the result of BiTE therapy. After that, coming to the clinical trials, the first clinical trial is safety and dosage form. So the first phase 1 clinical trial is subjected on or is performed on approximately 78 patients or volunteers. So the result was 70% of the volunteers are safe, but the adverse effects of those 76 members were fever and some hypersensitivity reactions. So moving forward, phase 2 clinical trial. In phase 2 clinical trial, 186 patients or volunteers were performed this BiTE therapy, and the result was the blimetamomab has given the most surprising and promising results. So after that, here comes the game changer, that is phase 3, and the phase 3 is named as Tower Trail. So what happened in phase 3? So almost 489 approximately volunteers were taken for the test, and they were divided into 2 groups. So one group is injected with normal chemotherapy, cytotoxic drugs, and another group was injected with blimetamomab, that is BiTE therapy drug. So the both results were almost same, but in chemotherapy, the people were becoming weaker day by day, but in BiTE therapy, the people were almost stronger and healthier, as we all discussed, like BiTE therapy only kills the cancer cells. In chemotherapy, the reason why people are very weak, because chemotherapy kills even healthier cells like all other which are useful for our body. And now comes to the pharmacokinetics. It has 2 hours of in pharmacokinetics, it has 2 hours of shelf life, which is very short, and it is metabolized, and the enzymes are divided into many more molecules. And after that, coming to the adverse effects. Adverse effects are little bit less compared to chemotherapy, like we get fever and neurological diseases like headache, nausea, vomiting, and other diseases like neutropenia, the reduction of neutrophils in our body which are related to white blood cells. And after that, the conclusion. So in my point of view, BiTE therapy is the most advanced therapy and all have to undergo that. But the main drawback or limitations for bite therapy is it is the most costliest therapy compared to chemotherapy. And it also has a limitation of accessibility, and the main drawback is cytokine release syndrome. So middle class people and above average middle class people can't afford it, but in my point of view, bite therapy is the most inviting therapy. Thank you so much.
In this valedictory function, let us start first by honoring the evaluators who have spent their valuable time for us today. First is N. Raghavendra Babu sir, Associate Professor from GRPC, College of Pharmacy. Saeed Ariful Hussain sir, Professor and Vice Principal, MESCO College of Pharmacy. Next we have Mr. Y. Srihari, G. Pullareddy College of Pharmacy. Dr. B. Narendra from Omega College of Pharmacy, please come on to the dais. Dr. S. Anuradha, Professor and HOD, SN Vanita Pharmacy Mahavidyalaya, please come on to the dais. Next, Dr. M. Lakshmi Chandani, Associate Professor and HOD, Omega College of Pharmacy. Ms. G. Shravani, Assistant Professor, Viper College. Please come on to the dais. Next, we go on to the winners of this poster and oral competition. First is the oral competition under the Department of Pharmacology. T. Meghanasri, MLR College of Pharmacy, has won the first prize. Second prize, Tanuja and Sangeeta from Viper. Please come on to the stage if you are available, or anyone from the college if they are available, please come on to the stage. Next, oral presentation in Department of Pharmacy Practice, Sania Begum from MNR College of Pharmacy, PharmD intern. B. Akshaya from MNR College of Pharmacy. Please come on to the dais. Oral presentation from Department of Pharmacognosy. First prize, Manogna from Viper College. And second prize, B. Swati from Gokaraju and Rangaraju College of Pharmacy. Oral presentation in Analysis Department, Sai Sandeep Verma from Viper College. Gokaraju, Rangaraju. First prize, ma'am. Second prize. Second prize. Manogna. First prize. First prize. Department of Analysis, Sai Sandeep Verma from Viper College and G. Manisha Reddy from GRCP. In the Department of Pharmaceutics, first prize for Priya Singh from Viper College. Priya Singh from Geetam University. Second prize, Pritika from GRCP. Next, we go on to the e-poster presentations. E-poster presentation, first prize. There are four, under each department. First one is Kirtana from Pradishta Institute of Pharmaceutical Sciences. First prize. Vishnupriya from Arya College of Pharmacy. Dhanush from Mallareddy College of Pharmacy. That one. Tasneem Fatima from MNR College of Pharmacy.
Now we move on to the second prizes of e-poster. First is Jasminta from Arya College of Pharmacy. Jugender from Mari Lakshman Reddy College of Pharmacy. M. Lokesh Kumar from Mari Lakshman Reddy College of Pharmacy. Now second prizes only. All second prizes. Jasminta from Arya College of Pharmacy. Jugendra and M. Lokesh Kumar from Mari Lakshman Reddy College of Pharmacy. Sania and Madiha from MNR College of Pharmacy. Requesting all the faculty to please come on to the dais.